Welcome student on another video lecture. Topic is biosynthetic pathway. In that we are going to study the biogenesis or biogenetic investigation, how the biosynthesis of particular compounds or different types of secondary metabolites, where they are synthesized, what is the site of their synthesis, and which type of enzymes are required and which pathway they will follow to synthesize that particular secondary metabolites, and how they are synthesized, that going to study by using different investigation studies. In that, in last lecture we have covered about the organ or cell, isolated organ or cell, and grafting method. Now another method is enzymatic study. So what are basically enzymes? Enzymes are the proteins. These are the protein molecules and they have a very important role in metabolism. Each and every reaction, it requires an enzyme. That enzyme will catalyze the reaction and it will convert from one form to another. And in this way, by using different steps or different methods, different pathway, they will synthesize the different types of secondary and primary metabolites. So to produce or to synthesize that secondary metabolites in the plant, it requires an enzyme. Without enzyme, the reaction will not occur, it will not catalyze, and it will not form secondary metabolites. So enzymatic study is also playing important role to study the investigation of biosynthetic pathway, which biosynthetic pathway the plant will follow to synthesize that particular enzyme or particular secondary metabolites, that can be studied by using enzymatic study. For example, phytochrome auxin plays critical role in regulation of plant growth and development. Auxin is a growth hormone in a plant and it will have an important role in regulation of plant growth and development. So indole 3 acetic acid is a major auxin that will be recognized in the, which is synthesized in the plant, but how that indole 3 acetic acid is actually synthesized in the plant, it is still unclear. So certain enzymatic studies, previous genetic and enzymatic studies have done to determine the synthesis of, that is growth hormone auxin or indole 3 acetic acid, where actually it will be synthesized and how it will be synthesized in the plant. So according to the previous genetic and enzymatic studies demonstrated that both tryptophan aminotransferase of Arabidopsis, that is TAA, and yucca flavin monooxygenase like protein are required for biosynthesis of indole 3 acetic acid. So to synthesize indole 3 acetic acid in plant, it requires tryptophan aminotransferase of Arabidopsis and yucca flavin monooxygenase like proteins. These are required for synthesis of indole 3 acetic acid. So this TAA family produces indole 3 pyruvic acid. So tryptophan aminotransferase Arabidopsis, it will produce indole 3 pyruvic acid, and the YUC family, yucca family, it will functions in conversion of that indole 3 pyruvic acid to indole acetic acid. So these both have a role in synthesis of indole 3 acetic acid, which is a auxin majorly found or recognized in the plant, which is a growth hormone. And how it will produce? It will produce by using these two, tryptophan aminotransferase of Arabidopsis TAA and yucca flavin monooxygenase protein. In that first one will produce indole 3-pyruvic acid, and that indole 3-pyruvic acid is converted into indole 3-acetic acid by Yucca family. It will helpful in conversion of indole 3-pyruvic acid to indole acetic acid, and in this way the auxin, that is indole 3-acetic acid, is synthesized in the plant, and the enzymatic study will helpful to investigate the biosynthesis of indole 3-acetic acid, which is a auxin growth hormone mainly found in the plant. Next one is mutant strains. Large number of mutant strains of microorganism has been produced. So whenever there will be some environmental changes or some physical or chemical agents are applied, or the lower plants like fungi or microorganism, when they are exposed to certain physical or chemical agent or UV radiation or some environmental changes, the mutant strains of these microorganism or fungi, they are produced. And when these mutant strains of these microorganisms they are produced, they lack in one or other enzyme. That particular enzyme which is required for metabolic pathway, so that enzyme is lacked in these mutant strains of these microorganisms like fungi or other small microorganism. And when that particular enzyme which is required for biosynthetic pathway or metabolic pathway is lacked, what will happen when that enzyme is not there, how the conversion will take place? It will result in blocking of their metabolism at particular stage. So that particular enzyme which is required for conversion into another form or to catalyze that particular reaction is not present, then what will happen? It will block the metabolism at particular stage. So metabolism will get blocked. When the metabolism will get blocked, what will happen? The intermediate which is produced before blocking of that metabolism, that will get accumulated. So now the metabolism is blocked because the enzyme is not present, it is lacking enzyme, and hence the intermediate which is produced, that will get accumulate before blocking. And so for its survival now it needs a artificial supply of another intermediate. So artificial supply of another intermediate is required, and when that artificially the another intermediate is supplied, then it will again for reinstallation of the pathway, biosynthetic pathway will take place, and it will synthesize that particular secondary metabolite. So such organisms are important for biosynthetic studies. So such mutant strains can be used in biosynthetic studies of natural product, because these mutants they are lacking certain enzyme, and due to lack of that enzyme it will block the metabolic pathway. Due to blocking of that metabolic pathway, the intermediate before blocking it will get accumulated, and to survive, for its survival it needs artificial supply of another intermediates, and due to which what will this microorganism, mutant strain microorganism are helpful in biosynthetic studies of natural product. For example, biogenetic pathway of gibberellins are mostly similar in both higher plants and Gibberella fujicorae. The mutant strain Gibberella can be used to obtain variety of novel C20 isoprenoid compound. So the biosynthetic pathway are gibberellins are mostly similar in higher plants. When we are considering the higher plant, the pathway which is used to produce gibberellins in the plant, that is near about same. The mutant strain of Gibberella can be used to obtain the mutant strain which contains Gibberella. It can be used to obtain variety of novel C20 isoprenoid compound, and which is produced at a level of geranyl phosphate in mevalonic acid pathway. We have seen the mevalonic acid pathway, and at the level of geranyl phosphate, these C20 isoprenoid compounds are produced due to that mutant strain, that is Gibberella. Very interesting results have been obtained by studies on ultraviolet induced strains of Claviceps purpurea. It is a fungus or ergot Claviceps purpurea, and studies are performed on that Claviceps purpurea. Interesting results are obtained on ultraviolet induced strains. So the ultraviolet radiations, that fungus will expose to ultraviolet radiation and it will produce a mutant strain. The mutant strain can produce amino acid of diverse nature. So such mutant strains of Claviceps purpurea, it will produce a amino acid of diverse nature when a ray plant is introduced with the spore culture of these mutant strains. Now the plant is introduced with the spore culture of these mutant strain. The sclerotia produced demonstrate the blockage of biosynthetic pathway of certain intermediates. So it will block when the mutant strains of the Claviceps purpurea they will use and they will the plant is ray plant is introduced with the spore culture of these Claviceps purpurea. Then what will happen? It will block the biosynthetic pathway for certain intermediate. It will block the biogenetic pathway of certain intermediate, and thereby the accumulation of specific alkaloid is also blocked. So the ergot alkaloid which are produced through that Claviceps purpurea, the formation or biosynthesis of that ergot alkaloid is get blocked due to that mutant strain. And the blockage after formation of chymoclavinine in mutant strain to such strain, if ergoclavin and other intermediates have been supplied artificially, then it will indicate the reinstallation of normal pathway and it will produce the specific alkaloid compound completely. So what will happen when a mutant strain is used? They will follow the different pathway and it will produce diverse nature of amino acid. And when a ray plant with such spore culture it will introduce, it will block the biosynthetic pathway for certain alkaloids, that is ergot alkaloid, which are the important alkaloidal compound from the drug, and it will get blocked. And when the blockage will occur due to chymoclavinine mutant strain, then if another intermediate, that is agroclavin, it will be supplied artificially, then again there will be the reinstallation of normal pathway will take place and it will produce the specific ergot alkaloid which is produced by that ergot drug. So in this way, this mutant strain can also be used to study various biosynthetic pathways of the drug, how the different secondary metabolites they can be synthesized. The investigation of these biosynthetic pathway can be done by using mutant strains also. So if you like my video, please press the like button and subscribe my channel. Thank you.